Chapter 9 introduces us to the physiology of the muscle tissues. Here we will specifically focus on skeletal muscle tissues and their function. So before we dive into the skeletal muscles, first let's begin by reviewing the characteristics of all of the muscle tissues. So the muscle tissues as a whole make up nearly half of the body's mass. All of the muscle tissues are specialized to transform the chemical energy of ATP that our body produces into mechanical energy, and that mechanical energy is in the form of movement. Now, muscles do this by contracting or shortening to produce pulling forces. So muscles produce movement by pulling on things. They can never push on things. In Chapter 4, we were introduced to the muscle tissues, so recall that we have voluntary and involuntary varieties as well. So let's continue on with the characteristics of the muscle tissues. So all muscle tissues contract or shorten to generate pulling forces, and the way that muscles do this is that structures inside the muscle cell actually get a little bit shorter, and that generates the pulling force of the entire muscle. And so that's how muscles generate pulling forces, and specifically we'll see that with skeletal muscles here in the next few lecture segments. So that's one characteristic of muscle, is that they are contractile. Muscles are also extensible and elastic. So muscle tissues are extensible, which means that they can stretch to a greater length. That is, when they are relaxed, they are able to be stretched by another muscle or another body part to a much greater length. Muscles are also elastic, or they have this property called elasticity, which means that they have the ability to recoil back to a resting length after they are stretched. So in this way of extensibility and elasticity, muscles behave a little bit like a rubber band, because we can stretch out a rubber band and it will recoil back to its resting length after it has been stretched. So in that way, we see some similarity between muscle tissues and the rubber bands that we're familiar with. So muscle tissue is also an excitable tissue, which means that it receives and responds to stimuli by changing its membrane voltage. So when muscles are not stimulated, that is, when they're at a state called rest, they return to a voltage called resting membrane voltage. This voltage is also referred to as resting membrane potential. So let's review resting membrane potential by first going back to this idea of selective permeability in the plasma membrane. So what you're seeing in this diagram here in the center is the plasma membrane of a cell. The extracellular fluid, or the outside of the cell, is on the top of the diagram, and the intracellular fluid, or the cytoplasm, is on the bottom of this diagram here. So recall from Chapter 3 that we learned of the selective permeability of the membrane, which means that the membrane is very specific about what has the ability to cross it. The small molecules that contribute to the change in membrane voltage of muscle cells are soluble in water. So this means that they have to diffuse into or out of the cell through these channel proteins. Now, these channel proteins provide a small water-filled tunnel for these water-soluble molecules to diffuse across the membrane. So remember that the inner core of our plasma membrane is hydrophobic. That means it doesn't like water. And so water and water-soluble substances do not easily move through the hydrophobic core of the plasma membrane unless they have a channel protein that helps them more easily get across the plasma membrane. So the main contributor to our resting membrane voltage is potassium. Now, resting membrane voltage is the voltage or charge that's measured on the inside of the cell at rest. That is, when the cell is not being stimulated here. So if we stuck a little voltage probe in here of the muscle cell, and the cell was at rest, we would read a negative voltage on the inside of the cell compared to the outside of the cell. So at rest, potassium concentrations tend to be higher on the inside of the cell than they are on the outside of the cell, which means that potassium is going to diffuse out of the cell through these potassium leak channels. So these potassium leak channels are just little water-filled channel proteins that aid in the passage of potassium across the plasma membrane. So potassium diffuses out of our cell through these potassium leak channels, and because potassium is positively charged, this means that the inside of the cell will become negatively charged when compared to the outside because these positive charges are leaving the cell. So as more potassium, more of that positive charge exits out of the cell across the plasma membrane, that's going to make the inside of our plasma membrane more negative with respect to the outside of the plasma membrane. So potassium actually leaves quite easily through those potassium leak channels, and the reason for that is that there are a lot of potassium leak channels in the plasma membrane. So it's very easy for potassium to find a doorway, if you will, out of the cell, because these little potassium leak channels behave as doorways for potassium to get out of the cell. And so because there's a lot of them, that means that it's very easy for potassium to get from the inside of the cell to the outside of the cell. We also have sodium leak channels, and sodium tends to leak into the cell very slowly through these sodium leak channels. At rest, what we see is that sodium concentrations tend to be higher on the outside of the cell than on the inside. So essentially the sodium concentrations are the opposite of the potassium concentrations, where we saw high potassium on the inside of the cell, we see high sodium on the outside of the cell at rest. The difference between the sodium and potassium leak channels, other than the substance that they're moving, is the number of them. So there's lots of potassium leak channels in the plasma membrane. There are very few sodium leak channels in the plasma membrane. So that means that sodium is not as easily able to get across the plasma membrane as potassium is. And so for that reason, we see a lot of potassium leaving, very little sodium able to get into the cell. And so that's what helps really contribute to our resting membrane potential that is negative, because we've got lots of positively charged potassium easily leaving our cell. So with potassium leaking out and sodium leaking in, the balance of potassium and sodium on each side of the membrane would be equal after a certain amount of time if it were not for the work of the sodium-potassium pump. And the sodium-potassium pump uses ATP to essentially help to recharge those concentrations of sodium and potassium on each side of the membrane. So what we're seeing here in this first step here is it's binding an ATP, and it's also binding 3 sodium from the inside of the cell. So what it's going to eventually do is going to pump these sodium ions from the inside of the cell to the outside of the cell. So the ATP that was bound to it is split. When ATP is split, that releases chemical energy and from the chemical bonds in ATP. And this changes the shape of the channel, the sodium-potassium pump, and expels these 3 sodium ions that were bound to it from the cell. So sodium is going to get kicked out of the cell. After these sodium are released, 2 potassium from the outside of the cell bind to the channel and will eventually be brought in. So in this next part of the diagram here, we see that the channel reverts back to its original shape, and the potassium that was bound to it is pumped into the cell. And so we see that the sodium-potassium pump uses 1 ATP to kick 3 sodium out of the cell and then bring 2 potassium into the cell. And so in this way, it's helping to keep the concentration of sodium on the outside of the cell quite high and keep the concentration of potassium on the inside of the cell high as well. So this diagram essentially summarizes the work of the resting membrane potential or voltage here. And we see that the potassium leak channels through which potassium leaves the cell are operating. We see the sodium leak channels, and that's allowing sodium to diffuse into the cell across the plasma membrane. And in the center of it all here, we see the sodium-potassium pump, which is maintaining those high concentrations of sodium on the outside of the cell, so that way sodium can continue to leak in. And the sodium-potassium pump is also maintaining high concentrations of potassium inside of the cell, so that way potassium can leak out and contribute to our resting membrane voltage.